Hi students, this is A. Mandari Kumar Rajesh. In this video, we are going to discuss another type of life cycle of phage called lysogenic cycle or avirulent cycle. In the lysogenic cycle, the phage DNA get integrated with the bacterial chromosome. The phage DNA get integrated with the bacterial chromosome and it can multiply along with the bacterial chromosome. The phage DNA can multiply along with the bacterial chromosome. Then no virus particles are produced independently. In the lysogenic cycle, no virus particles are formed independently. So you cannot find any virus particles are formed independently in the lysogenic cycle. Then I am going to explain the whole process one by one. In the first step, the bacteriophage get attached to the bacterial cell wall and it injects linear DNA. It injects linear DNA into the host cell. After that injection, the linear DNA become circular. The linear DNA become circular and get integrated with the bacterial chromosome. Get integrated with the bacterial chromosome by the process recombination. By the process recombination. Then the integrated phage DNA is now called as prophage. The integrated phage DNA now called as prophage. What is called prophage? What is meant by prophage? Pro means before. Pro means before. So the prophage means the state of a virus in the form of genome. In the form of genome get integrated to the host DNA. Get integrated to the host DNA before being activated inside the host cell. Before being activated inside the host cell, and that condition is called as prophage. I once again repeat the definition. Prophage means the stage of a virus in the form of genome. In the form of genome get integrated with the host DNA. Get integrated with the host DNA before being activated inside the host cell. That means I already told you earlier, lysogenic cycle or avirulent cycle. So in this life cycle, the bacteriophages are avirulent. The bacteriophages is avirulent. So they are in inactivated form in the lysogenic cycle, and that stage is called as, and that condition is called as prophage. And that condition is called as prophage. Then the prophage activities are controlled by the prophage activities are controlled by two repressor proteins. These prophage activities are controlled by two repressor proteins. Repressor proteins produced by phage genes. Produced by phage genes. Why the phage genes has to produce the repressor proteins to control the prophage activity? So the nature of bacteriophage is it has to multiply inside the host cell and it takes over the biosynthetic, that is the host cell machineries, and it controls the metabolism of the host cell and it disintegrates the bacterial chromosome. But that condition is not happen in this life cycle because of the presence of the repressor protein. So these two repressor proteins controls the activity of prophage. Controls the activity of prophage. Actually, repressor proteins binds with DNA or RNA. Binds with DNA or RNA, and it can inhibit, this protein can inhibits the expression of phage gene. It can inhibits the expression of phage gene. So with the help of the repressor proteins, the prophage activities are suppressed. That is why the prophage are inactivated in the lysogenic life cycle. Because of the repressor protein, the prophage is inactivated in the lysogenic cycle. Then, however, the bacterial cell undergone the cell division. At the time, the phage DNA also multiply along with the bacterial chromosome. When the bacterial cell exposed to the UV radiation or the chemicals, at that time excision of DNA takes place. Excision of DNA takes place and they can multiply inside the host cell and they can released from the host cell by the process lysis. So that's all about the lysogenic cycle or avirulent cycle. Now I am going to tell about some important terms like virions, virusoids, viroids, prions and types of phages. So please wait a minute. Let me tell about the difference between the following terms. The first term is virion, then second one is viroid, the third one is virusoids and the fourth one is prions. Now I am going to explain each term one by one. First I go with virions. Virions are nothing but virus. Virions are nothing but virus. That means they are complete infectious virus particle and they are present outside the host cell. That is the only difference. When it is inside the host cell, they are called as virus. When they are outside the host cell, they are called as virions. That is the difference between virus and virions. If that infectious particle present inside the host cell, they are called as virus. The infectious particles are present outside the host cell, they are called as virions. That is the difference between virions and virus. Then the next term, viroid. Next term, viroid. Viroid means virus like. Viroid means virus like. Then the viroid was discovered by the scientist Theodor Diener in the year 1971. In the year 1971. So viroid means virus like. Viroid means virus like. Then what is the difference between the virus and viroids? Virus has the protein coat. Virus has the protein coat which encapsulated the DNA. While in the case of viroids, the genome is not encapsulated by the protein coat. So the RNA is not encapsulated by the protein coat. That is the major difference between viroid and virus. Viroid and virus. So viroid contains single-stranded RNA. Viroid contains single-stranded RNA and they do not have the protein coat. Then that RNA molecular weight is low. So the RNA has low molecular weight. The RNA has low molecular weight. Then this single strand RNA can self-replicate. This single strand RNA can self-replicate like the virus. Self-replicate like the virus. Then these viroids can cause disease in potato. The disease name is potato tuber spindle disease and citrus exarctis. So these viroids can cause serious damages to the potato plant and the citrus plant. It causes commercial losses. It causes serious commercial losses. So the difference between virus and viroid is virus contains the protein coat. The virus contains the protein coat which encapsulated the DNA or RNA, while in the case of Viroid, they do not have the protein coat. They do not have the protein coat. Then the third term is Virusoid. The third term is Virusoid, and it was discovered by the scientist J.W. Randalls and co-workers and their co-workers in the year 1981. Then what is the difference between Virusoids and Viroid? So Virusoid contains the genetic material single-stranded RNAs. Single-stranded RNAs like Viroids, but Virusoids cannot self-replicate. They are not able to self-replicate. So that RNA cannot able to self-replicate. So that is the major difference between Virusoids and Viroids. So Virusoid cannot able to self-replicate. It requires, this Virusoids requires, I am going to write that point here. So please listen. So the Virusoids requires helper virus. So the Virusoid requires helper virus because the single-stranded RNA, that the genome cannot able to encode for a protein. So it requires helper virus to enter into the host cell. After the entry of the helper virus, the Virusoids are actively function inside that host cell, and they produce the genome. They produce the genome by the process replication. For the replication of the genetic material, it requires the helper virus. That is the major difference between the Viroid and Virusoids. Then Virusoids also can cause serious damages to the commercial crops. And it leads to serious economical loss. Then the final term is Prions. Generally, the scientist, and as we are a science student, isn't it? As a science student, we believe that any infectious particle contains DNA or RNA as the genetic material. But in the case of Prions, they are proteinaceous infectious particles. Proteinaceous infectious particles are Prions. So the scientist and we believe that the infectious particles must contain DNA or RNA as the genetic material, but it is a protein. It can infect, it can cause disease to the plants as well as the animals. So this Prions can cause disease to the animals and plants. That is why the Prions are otherwise called as proteinaceous infectious particle. Then the Prions are very, very dangerous, and it was discovered by Stanley B. Prisoner in the year 1982. Prions are very, very dangerous because there is no medicine to treat the disease which is caused by the Prions. Mainly the Prions which affects the brain. It makes the brain like the spongiform lesions. So there is no medicine to treat that type of disease, the type of disease. So the prions are very dangerous. There is no medicine for the disease which is caused by the prions. Mainly these prions are developed from the contaminated meat, and from some animals, that not properly maintained animals. So we have to avoid that type of contaminated meat and not properly maintained animals. So that is the only way we can escape from the prions. Otherwise the prions may get affected the human beings. So please have or consume the meat or any kind of non-veg items with a proper cooking machineries, proper cooking procedures. Otherwise don't consume that meat because it may contain prions and it can cause serious diseases. So we cannot find solution for the disease which was caused by the prions. Then I am going to tell about some of the viral disease which is caused in the animals, the human beings and the plants. Here I have written some of the disease which is caused by the virus in plants, animals and humans. So in plants the virus can cause tobacco mosaic disease, cauliflower mosaic disease, sugarcane mosaic disease, potato leaf roll, bungee top of banana, leaf curl of papaya, vein clearing of lady's finger, rice jingro disease, cucumber mosaic disease, tomato mosaic disease. So how can we prevent this type of plant disease? There is no proper medicines to treat the plant disease for any kind of viral disease. So for the plant disease we have to select the virus free plants. If the plants are affected by this virus, we have to totally ban that area and we have to quarantine every time before planting the commercial crops. So that is the only way we prevent the plants from the virus. Then animal diseases like foot and mouth disease of cattle, rabies of dog, encephalomyelitis of horse. That means encephalo means brain. It affects the brain of horse. Then human disease like common cold, hepatitis B, cancer, SARS, AIDS, rabies, mumps, polio, chikungunya, smallpox, chickenpox. So these two type of diseases are eradicated by vaccination. Then measles. So these diseases are caused by virus. So we can escape from this disease by making our body health and hygiene. So we have to maintain proper hygiene. So we are not have any kind of that illegal activities or any unwanted consuming some unwanted things. So we have to avoid these kind of unwanted things, then only we can escape from these kinds of disease. So we should make our body clean and healthy. So we have to maintain proper hygiene. So by that method only we can escape from this type of disease. Otherwise that virus cause serious damages to our body. There is no proper medicine to treat the viral disease because the major defect is virus do not have the cell wall. While in the case of bacteria it has cell wall, so we can treat the bacterial disease by applying antibiotics. But in the case of virus, they do not have cell wall and they do not have any proper cell machineries. So Due to that reason, we cannot treat the viral disease easily. Then I am going to tell about some of the different kinds of phages. The virus which affects the blue-green algae, they are called as cyanophages. Then the virus which affects the bacteria is called as bacteriophages. Then the virus which affects the yeast, they are called as cymophages. Then the virus which affects the fungi, they are called as mycophages. The virus which affects the fungi is called as mycophages. Then the virus which affects the animal is called as zoophages. The virus which affects the animals are called as zoophages. Then the virus which affects the plant is called as phytophages. So viruses are the serious entities which can cause serious diseases. So please stay away from this kind of particles and ensure the proper hygiene and health. So that's all about today's video. If you have any doubt, please clarify with me. Thank you very much for watching this video. Thank you.